Okay, so we just finished explaining PCR and the overall process of PCR. And then just to let you know, which we'll do in the tutorial, that there is there are many different tools that you can use to design primers, one of which is Primer Blast at the NCBI. So you input your sequence, and then you can either input some primers that you have, and it can predict how well they will work, or you can ask it to predict primers by choosing different parameters, the size that you want, approximate positions that you want them, melting temperatures, etc. And there it has information about the organism, so it can look up and search to see if those primers are going to hit anywhere else in the genome. So this is an example. So it predicts potential primers, the length, the needing temperature, and where it hits, and then if it is likely to hit somewhere else in the genome. So as I said, we'll go through that in the tutorial as well. So moving now on to gene prediction. So a gene is really a region of a genomic sequence that corresponds to a unit of inheritance. It's usually associated with regulatory regions, transcribed regions, and other functional sequence regions. So you now get non-coding genes, but what I'm focusing on here is looking at protein coding genes. I'm not going to go into gene prediction across whole genomes in detail. I'm just going to give you a basic idea of how you'd find a coding sequence and look for potential other signals. So gene prediction relies on a number of different steps. So you want to look for gene structures, which would be your coding sequence and your regulatory features. And so these tools tend to just go along the sequence looking for coding regions and then intergenic regions and then features on either side of the coding region. What they would also do is check the reading frame. So I'll describe what I mean by reading frame next. It looks for other features like promoters and polyadenylation signals. So poly-A tails tend to be at the end of genes in eukaryotes. And then in eukaryotes, of course, there's a lot more complexity because you have introns and exons. And so clever gene prediction tools will be able to identify these. It's important to use EST or BLAST support. So sometimes you don't know if it's really a real gene. So EST is an express sequence tag, and so you blast your predicted region against an EST database to see, has the transcript ever been reported and submitted to a database? In other words, is that piece actually known to be transcribed? And this is very useful for reading out potential false gene predictions. So translation is really where you want to say, here's a piece of DNA. Obviously it goes via RNA, but from the RNA, what protein does it make? And this is all determined by the actual DNA sequence. So if you know which frame it's in, you can select that when you're doing translation, or you can do a 6-frame translation. And I'll describe what I mean by that shortly. Usually genes start with an ATG, so we usually choose the start codon of ATG, although there are known exceptions. And then the program will do a translation using the genetic code and then look for stop codons, which will indicate the end of a gene. So ATG starts, and then there are particular stop codons which stop the gene. So usually in the results, the longest ORF starting with the methionine, which is what ATG encodes, and ending in a stop codon with no stop codons in the middle should be a real protein. And then, as I said, you can confirm this by looking at other regions upstream or downstream of the EST evidence. So it's important also to use an appropriate codon usage table. So different organisms, because they have different GC contents and different transcription and translation machineries, they have a slightly different codon usage table. So an open reading frame, so an ORF, is basically a string of in-frame combinations or triplets of bases that specify an amino acid. So an amino acid is specified by 3 bases in the DNA sequence. As I've said, an ORF will start with a start codon and end with a stop codon. If you just have one base inserted or one base deleted somewhere in the middle of that, suddenly that triplet, that sets of 3 bases, is different after that insertion or deletion. So that will be called a frame shift. So when I say that you have different codon usages and you have these 3 bases encoding a particular amino acid, this is what's known as the genetic code. So you is replaces T in RNA. So in RNA it's U, whereas in DNA it would just be the T. So this could equally be replaced by T if you're looking for DNA. Now there are multiple different sets of codons or triplets that make up a particular, that would correspond to a particular amino acid. So in some cases here, you'll see that there are 4 different possible triplets that will encode serine. What you'll see is that the first 2 bases are very conserved, and the third base is flexible, pretty much anything. So that's important. So if you get a mutation in the first 2, it's quite likely to change the amino acid. If you get a mutation in the last one, you'll still end up with the same amino acid. So now we have 4 bases, A, C, G, and T, so there's 64 possible codons. Now actually, there's 61 possible codons that lead to an amino acid, and the last ones actually lead to a stop codon. So these are stop codons here. If you find these in the sequence, that represents the end of the coding sequence. And then AUG, which would be ATG, is the start codon. So when you come to translating sequences, basically these programs use that table, it uses the genetic code. Now the reason we say that there are 6 possible reading frames is because there are 3 in each direction. So if you look at the forward strand, this is from 5' to 3'. This is the reverse strand, the reverse complement, which is 3' to 5', and the opposite. So it's reverse, so A would be, the opposite is T, A's opposite is T, G is C, etc. So you've just taken the strand, you've got the complementary sequence, and you flipped it around, so it's always 5' to 3'. So now you go along looking for your first, and your frame 1 is starting from the very first base. This would encode those, but of course you haven't got a start codon of ATG yet. So this would be frame +1, which is basically looking at the triplets starting from frame 1. And there it's simply a lookup to go back to this table to say which amino acid is encoded by those 3 bases. But then you could also have genes being transcribed on the reverse strand. So you have to look at that, because now the sequence is actually a bit different. AAG will give you a different amino acid to AGT. So this will be looking at the -1 direction. So frame +1 and frame -1. So we're starting from the first base, but on the forward, the reverse strand. If you decided to skip the first base and start on the second base, this now becomes frame 2. And you can see now that they're very different amino acids that are coming in. And in fact, this immediately encodes a stop codon, so it can't be frame 2, because you would end your protein pretty much straight after you started it. And then equally, you can start from the second base to get frame -2. If you move now, you skip the first 2 and you start on the third one, that is frame +3. Start on the third one on the reverse complement, that's now frame -3. So this is how you get 6 frames. Frame 1, 2, and 3 on the first strand, -1, -2, and -3 on the reverse complement strand. So you have 6 possible frames, each of which will lead to a different set of amino acids. Some of which will actually encode a protein, some of which will never actually be made into a protein. So we do 6 frame translation. If you then moved to the next base, you leave the first 3, you'll see that in fact this triplet becomes the same as the first one. So it's the same frame because you've now shifted by 3 bases. So that's why there's just the 3 frames. So when you get the final protein, you do 6 frame translation, and it gets all these different fake little products, and usually the longest one in bacteria, starting with a stop codon and ending with a stop codon, is what you want. So gene prediction in bacteria is relatively simple because you have this nice ATG, you have your stop codon, you have a reasonably well conserved ribosome binding site at a particular position, and then the promoter region has a -10, so 10 bases upstream of the start, and -35, so 35 bases upstream of the start. They're quite reasonably well conserved pieces of DNA. So it's reasonably easy to predict genes in bacteria. When it comes to eukaryotes, however, you have introns and exons. And so it's difficult to find little introns when you have exons, when you have these huge introns in between. So you'd need to find features like splice sites, splice junctions, and confirm the pieces of the exon from EST evidence. So what happens at alternative splicing is the intron jumps gets cut out and the exons get stuck together. But then, of course, you also have alternative splicing where different exons can be brought together to make different products. So even if you can predict the protein sequence within each exon, you don't necessarily know the final combination of exons. You would need EST evidence, again, for this. If you look at a transcript in ensemble, you'll see the relative size. So the green is the huge intron and the red is the small exon. So exons are very small. This is in a human genome. Exons are very small compared to introns. So it's really like looking for a needle in a haystack, which is why gene prediction in humans is challenging. So what the prediction programs tend to do is they'll find features, sometimes using position-specific scoring matrices. You probably hear about that in later modules. You can look for splice sites, stop sites, and then you can use these and various other signals to predict exons. And then they might have specific compositional biases, so percentage GC content. You can also check by looking at homology information. So has this protein that you think you're making been seen in another organism? That will also be evidence that that is the correct protein. So you get these different bits of evidence, you put them together to try and work out exactly where pieces of your gene are. Some methods use hidden Markov models, so where these features are actually features and states. And then obviously many of them use EST information to confirm that is the transcript that you have predicted. Just to note that if you were aligning an EST sequence against a genomic sequence, you'd have these big gaps in the middle because they would only align at exons, because the EST is a piece of transcript where the introns have already been removed. So there are various different gene prediction software tools. You can try some of them. We're not going to go into detail. It's normally if you're going to be doing large-scale gene prediction for whole genomes. Then you also have promoter prediction tools, and promoter prediction is very challenging because although in bacteria some are conserved, it is actually very difficult to find promoter regions and evidence of these. But there are some prediction tools that will try to predict where there might be promoter sequences. So that's DNA sequence analysis. So there are various different features you can find, including genes, but there's a lot of complexity about, some are more reliable than others because the signals are more conserved. Now, RNA sequence analysis, I'm just going to mention in this one slide, there are many different types of RNA. So you get tRNAs, rRNAs, mRNAs. So the transcript that comes from a gene would be your mRNA. Some mRNAs have their own activities, like ribozymes, and some prediction programs you can see to see how RNA folds, perhaps into a ribosome-like structure. And now with the new field of small non-coding RNAs and microRNAs, there are a number of tools that have emerged for identifying potential non-coding RNAs and the targets that they bind to. And we can do that because there are certain features of microRNAs, like how they fold, free energy, etc. And then the complementarity of that allows us to find the targets. So secondary structure is very important, and there are many tools for looking at secondary structure of RNA. And then, of course, the obvious analysis for RNA would be gene expression analysis, which is a whole new, a whole different kind of analysis, which I don't have time to go into here. So to summarize, basic sequence analysis is about finding features in a sequence. So it could be small things like just finding restriction enzyme sites or designing primers for cloning PCR. Or you can look for bigger signals and combinations of signals to enable you to do something like gene prediction. But in most cases, what it is, we're just looking for pattern matching, because we know that certain patterns are representing restriction enzyme sites, promoter regions, transcription factor binding sites, etc. Which is why bioinformatics is useful in finding features, because it's looking for these patterns in a sequence, and computers can do that. So that is the end of DNA sequence analysis, and there's a tutorial that will help you to play around with some of these tools. Thank you.